Human reproduction operates on a strict, sequential timeline. The process relies on defined anatomical waypoints, starting with gametogenesis and the cyclic release of the ovum. The World Health Organization defines reproductive health as a total well-being in all aspects of reproduction. This encompasses physical, emotional, behavioral, and social health. Achieving this baseline requires coordinated societal effort. India initiated national family planning programs as early as 1951 to address population growth and reproductive health. The earliest steps in these programs prioritized sex education in schools. Building foundational awareness was deemed essential to prevent abuse and reduce sex-related crimes. At the biological level, the human reproductive sequence passes through 4 major waypoints: gametogenesis, fertilization, cleavage, and implantation. Modern clinical intervention manages human reproduction by intentionally targeting these specific waypoints, either by blocking them to prevent conception or surgically bypassing them to assist it. Reaching the WHO's goal of reproductive well-being requires us to engineer this biological timeline with medical precision. The most reliable method to prevent pregnancy is to interrupt the timeline early. By targeting the pituitary-ovarian axis, interventions can stop ovulation before an ovum is ever released. This graph tracks lactational amenorrhea. Intense postpartum nursing generates high prolactin, suppressing LH and FSH, which prevents ovulation. However, around the 6-month mark, nursing frequency decreases, the prolactin curve dips, and ovulation can resume unpredictably. To extend this type of suppression reliably, medicine relies on artificial mimics. Oral contraceptives deliver small, precise doses of progestogens or synthetic progestogen-estrogen combinations. These synthetic hormones maintain the suppression of the pituitary gland, inhibiting both ovulation and uterine implantation. Manipulating the endocrine feedback loop remains the primary strategy for restricting gamete availability long before fertilization poses a risk. If an ovum is released, the intervention strategy shifts to the next node on the timeline, establishing a physical or chemical blockade against sperm. Traditional barrier methods, including condoms, diaphragms, and cervical caps, physically prevent gametes from meeting within the reproductive tract. Condoms serve a vital secondary function in public health. They protect users from sexually transmitted diseases, ranging from syphilis and hepatitis B to HIV. Limiting the spread of reproductive tract infections relies on individuals avoiding unknown partners and seeking immediate medical consultation when symptoms arise. Beyond simple physical barriers, intrauterine devices, or IUDs, chemically alter the internal environment of the uterus to intercept gametes. This schematic demonstrates a copper-releasing IUD, such as the CUT. Once inserted, the device continuously disperses copper ions into the uterine cavity. These copper ions act as potent spermicides. They drastically suppress sperm motility and strip away the cell's fertilizing capacity. The presence of the device and the altered chemical environment also trigger a massive local immune response. White blood cells rapidly activate within the uterus. They initiate aggressive phagocytosis, actively engulfing and neutralizing any incoming male gametes. Barrier and IUD methods succeed by weaponizing the anatomical pathway itself, turning natural transport routes into hostile environments. Biological theory alone cannot dictate policy. Managing population-level reproductive health requires strict statistical quantification of how often a method fails. The Pearl Index measures contraceptive efficacy. Testing a pill on 500 women for 2 years yields 1,000 women years of exposure. With 10 pregnancies, we multiply by 100 for a 1,000 numerator. The result is a Pearl Index of 1, a 1% annual failure rate. Clinical infertility is defined as the inability to conceive after 2 years of unprotected sexual cohabitation. Treating it requires a shift from blocking the timeline to reconstructing it. When natural pathways fail, clinicians utilize assisted reproductive technologies. These involve specialized cellular and surgical bypasses. This AOT decision tree splits fertilization into in vivo, inside the body, or in vitro, outside. For in vivo methods like GIFT, an unfertilized ovum is transferred directly into a fallopian tube. Conversely, in vitro fertilization isolates gametes to fertilize in laboratory conditions. Once fertilized outside the body within the laboratory, The newly formed zygote undergoes rapid cellular division, multiplying precisely into early-stage blastomeres. The destination for embryo transfer is dictated by the 8 blastomeres threshold. Zygote intrafallopian transfer, or ZIFT, routes early embryos with fewer than 8 blastomeres back to the fallopian tube. Embryos developing past 8 blastomeres require intrauterine transfer directly into the uterus. ART functions as an architectural workaround. Clinicians select the precise biological waypoint at which to reinsert the embryo based entirely on its developmental maturity. Once gestation begins, the focus of reproductive health shifts from pathway engineering to diagnostic monitoring. This visual illustrates amniocentesis. A needle samples the amniotic fluid, allowing doctors to extract fetal cells and map the chromosomal pattern. Because this karyotype reveals sex chromosomes, the procedure is banned for sex determination to combat female feticide. Despite this restriction, the test is legally and medically essential for high-risk pregnancies. It screens for lethal genetic abnormalities and chromosomal defects, including hemophilia, sickle cell anemia, and Down syndrome. If severe abnormalities are detected, clinicians may advise medical termination of pregnancy before full term. MTP is clinically necessary in scenarios where the continuation of pregnancy is fatal or severely harmful to either the mother or the fetus. However, this intervention has a strict physiological safety window. MTP is considered relatively safe only during the first trimester, up to 12 weeks of pregnancy. True reproductive health requires the active, evidence-based management of human biology, from the earliest stages of gametogenesis all the way through gestation.